Hello, good evening to all. Today we will discuss regarding to the collagens, and as we know, collagen is a connective tissue and one of the most important connective tissue, and it comes under the ECM, extracellular matrix. We have seen the few points regarding to collagens also. Now here we will discuss regarding to collagens. First of all, you see structure and composition of collagen. Already you know collagen molecule is what type? It is a fibrous protein. Most of the extracellular matrix components are the protein, and among the all, one is collagen is very important, and it is what type of protein? Fibrous protein, you see. And here you see collagen molecule is fibrous protein. What type of protein? Fibrous protein. You see, collagen molecule is a fibrous protein, has an elongated triple helical structure. This point is very important. What type of structure? Triple helical structure. Underline it. You see, triple helical structure. Triple helical structure. It's very important. You must be. So if it is a fibrous protein, it means it is made up of amino acids. And what are the rich amino acids of this one is proline and glycine. We can say the collagen mainly rich in proline and glycine amino acids. Though they are made up of so many amino acids, but rich in proline and glycine. Proline is responsible to create helical structure. क्या काम है proline का? The function of proline is to create the helical structure. कहने का मतलब ये है कि proline helical structure बनाने में help करता है. And glycine is found in every third position of the polypeptide chain, and it is studied that the glycine, which is one of the simplest and basic amino acid, it is found every third position of the polypeptide chain, as it is given glycine XY. Third position it will be there. Now we will see metabolism of collagens. If we are saying the metabolism, it means we are talking is about of anabolism as well as catabolism both. Anabolism mean synthetic part and catabolism mean degradative part. So metabolism, if you are saying metabolism, it means we have to discuss is about of anabolism as well as catabolism both. Now here first we will see the anabolic part of this one, and anabolic part is mean biosynthesis of collagen. If someone is asking what is the anabolic part of the collagen, that is the biosynthesis of the collagen. You see, biosynthesis of the collagen. Biosynthesis of the collagen. Here. Now, if biosynthesis will take place, it means what? There is need of precursor, and no doubt precursor will be some particular type of biomolecules or cells. Here, fibroblasts Fibroblasts are the precursor for the synthesis of the collagen. And if you are saying fibroblasts, then here two content. One is osteoblast, which is arise from the bone, and another is chondroblast, which is arise from the cartilage. Cartilage is a source for chondroblast cells, and the bone is a source for the osteoblast cell. These are the precursors which will be responsible for the formation of the polypeptide of the collagen. So, with the help of osteoblasts and chondroblasts, there will be the formation of first stage of it, that is called pre-procollagen. Pre-procollagen means alpha chains of polypeptide precursor. Alpha chains of polypeptide precursor. As we know, it is a polypeptide, so there is presence of alpha chain. So pre-procollagen is alpha chains of polypeptide precursor. Its main osteoblasts and chondroblasts will be responsible for the formation of the pre-procollagen. Now here, pre-procollagen converts into pro-alpha chain through the processing of ribosome and RER. So pre-procollagen will convert into what? Pro-alpha chain. Osteoblast to pre-procollagen, pre-procollagen to pro-alpha chain. And this pro-alpha chain will form with the help of the processing of the ribosomes and RER. RER, rough endoplasmic reticulum. Rough endoplasmic reticulum. So there will be a lot of processes by the ribosomes and rough endoplasmic reticulum. This pre-procollagen will convert into the pro-alpha chain. Now pro-alpha chain processed by so many enzymes. There will be the processing, so many enzymes through the hydroxylation and glycosylation. Now here two terms is hydroxylation and glycosylation. Hydroxylation, you can say the addition of OH group, and glycosylation, addition of the some carbohydrate moiety. So pro-alpha chain will be processed with the help of enzymes through the processes of the hydroxylation and glycosylation, and it will convert into the procollagen. Now this procollagen molecules convert into mature triple helical molecule. Procollagen, these procollagens combine to each other and they will form the triple helical molecule under post-translational modification. So before the formation of the triple helical molecule of collagen, there will be the post-translational modification, and after post-translational modification it will make this type of molecule, that means triple helical molecule. Post-translational modification means the modifications which are happening after the synthesis of the protein structure. As you know, during the in genetics process, there are central molecular dogma, such as the first replication, formation of DNA to DNA is replication, and DNA to RNA is transcription, and RNA to formation of a particular polypeptide, peptide or protein is called translation. And after translation, there will be the few modifications in molecules that is referred as post-translational modification. So hydroxylation, glycosylation, methylation, you can say deletion of few parts. All these parts are the post-translational modification. Clear? Now, mature collagen molecules mature collagen molecules spontaneously associate to form fibrils. Okay, ये क्या करेंगे, जो मेच्योर कोलेजन मॉलिक्यूल हो जाता है वो स्पॉन्टेनियसली स्वतः अपने आप एक दूसरे से एसोसिएट होकर के क्या बना देते हैं, फाइब्रिल्स बना देते हैं. राइट, दे फॉर्म्स द फाइब्रिल्स. जैसे हियर collagens with cross linking convert into tropocollagen. So these are the fibrils. There will be the cross linking. The fibrils will form the collagen. Now collagen with cross linking, sometimes these collagens make the cross linking to each other, and the term is used tropocollagen. Tropocollagen is cross linked collagens. Tropocollagen is a cross linked collagen. Cross linked collagen. What type of tropocollagen? If there is, what do we mean of tropocollagen? Then answer must be, we can say tropocollagens are the cross linked collagens. Cross linked collagens are referred as tropocollagen. Right? Now, if you want to see the biosynthetic process of this one in diagram, I want to focus it in this and try to understand. You see here structure is given. You see, now DNA structure is here. DNA structure is there. You see DNA. The whole, this particular thing, it is a cell. Sorry, this particular is nucleus. You can suppose this particular total is a cell, and in cell presence of the nucleus. In nucleus there is presence of the DNA structure. Now in this DNA structure there is a particular gene which will be responsible for the formation of the proteins regarding to the collagen. Here you see in nucleus one is COL gene. COL gene is responsible for the formation of the messenger RNA which is required for the formation of the proteins or polypeptides in collagen. Now this COL gene, COL gene will be responsible formation of the messenger RNA through the process of the transcription. You hear one term is transcription. So this term has been taken from the transcription, and generally here in molecular biology or molecular diagram, transcription is a formation of the RNA from the DNA. Thing is this, it is a formation of the RNA from the DNA. Now here you see messenger RNA. So messenger RNA is forming inside the nucleus. transcription is a process. It is a process which are happening into the nucleus. But after the formation of the messenger RNA, this messenger RNA will enter into the cytoplasm of the cell. And in cytoplasm there is translatory machineries, or you can say translatory components. These translatory components will combine with the messenger RNA of this one, and they will be responsible for the formation of the proteins regarding to the collagen. In translatory machineries there are so many components, but you can say ribosome is one of the most important component which will provide the platform for the ligation of the messenger RNA. And so many factors will be there in cytoplasm which will participate into the formation of the translatory into the formation of the proteins, or we can say translatory processes. So in cytoplasm, when transcription process is happening into the nucleus, while translational process or translation process will happen into the cytoplasm of the cell. Why? Because all the components, all the things which is required for the formation of the protein or polypeptide chain present into the you can say here. That's why it's happening into the cytoplasm. Now here mainly we will see, here you see, yeah, one point. Okay, here is in messenger RNA you see this single peptide bond on left side, extension peptide on left side. Right side you see extension peptide is there. Now this messenger RNA will change into the polypeptide chain through the translation process, and in this polypeptide chain, that is the pre-pro-collagen, there is presence of extension peptide, signal peptide, and these external and signal peptides will be removed with the help of few enzymes. And here you see that is we can say post-translational modification. Mean pre-pro-collagen under the post-translational modification will change into the pro-collagen. What are those translational modification you see? There is a such as here intracellular modifications, that is the you can say post-translational removal of signal peptide, hydroxylation of proline and lysine, clear, with the help of enzyme prolyl hydroxylase and lysyl hydroxylase. In presence of vitamin C, oxygen and Fe2+. You see, one point is very important to note here. The hydroxylation of the proline as well as lysine mainly takes place in the presence of vitamin C. So the importance of vitamin C is very much, because if vitamin C will be not there, then the mature collagen will not form, and it will cause the disorders regarded to the you can say gum bleeding, etc., and that is referred as scurvy. Now glycosylation and triple helix formation mean in post-translational modification these things will take place, and it will change in pre-pro-collagen will change into the pro-collagen. Now pro-collagen through the exocytosis process. What do you mean of exocytosis process? Exo mean bahar, cytosis cytosis term has been taken from the cell. So if anything is releasing out of the cell, then it is referred as exocytosis. You see, exocytosis. Extracellular modifications. So extracellular modifications, One was the post-translational modification, another was the exocytes. सेल से बाहर आ गया कौन? प्रोकोलेजन. Now, extracellular modification will lead to the mature procollagen, tropocollagen. Here you see removal of extension peptides, oxidation of amino group of lysine to aldehyde by lysyl oxidase in presence of vitamin C, oxygen and copper. Cross-linking of triple helix by covalent bonds lead to the mature tropocollagen. You see, it was, you can say here, it was your biosynthetic part of the collagen, which is, that one is diagrammatic portion, and it's very important. In this way, if we will show, we will get So that was the biosynthetic part. I think that it is sufficient for today, and after that we will see the another part of the collagen metabolism, that is the degradative part, or you can say catabolic part. Thank you very much.